So far, nothing I have said about evolution should seem surprising or controversial. This is because we have focused on microevolution, that is, short-term changes within species. The controversy arises when we infer that if small changes can occur over a few generations, then large changes must occur over millions of years, and when combined with reproductive isolation, leads to the formation of new species. This is macroevolution. It is not directly observable because it takes more time than the time humans have written history for. If we are going to talk about what separates species, we need to start out by defining what a species is. Species generally look similar to one another, but where do you draw the line? The butterflies on the left are considered different species, but the ones on the right are all considered one species with phenotypic variation. Are the patterns similar because of convergence, or the fact that their ancestor had genes for these patterns? We know that they are different species because the ones on the left are genetically divergent, whereas the ones on the right are genetically similar. But sometimes genes can evolve slowly, and you can end up with different looking populations separated geographically for long periods that seem to be different species, but have indistinguishable DNA. It is very subjective. But ornithologist Ernest Mayr made a suggestion: define species based on whether or not they can successfully produce viable offspring. He called this the biological species concept. It usually works pretty well. If a species has been split for long enough, differences in their biology prevent them from being able to produce fertile offspring. Even if a giraffe attempts to mate with a donkey, the sperm and egg cells are incompatible. In order for speciation to occur, you need some sort of mating barrier. We break these forms of reproductive isolation into two types: pre- and post-zygotic, referring to whether they occur before or after mating. Pre-zygotic isolation can be caused by geographical barriers like mountains or rivers, having different mating seasons, responding to different courtship behaviors, or an inability to physically have sex, though that giraffe gets an A for effort. Which of the following is an example of temporal isolation? Post-zygotic isolation occurs after mating. Gametes may be incompatible, or maybe fertilization occurs, but the hybrid offspring don't live very long because of developmental deformities associated with genetic differences. It is like mixing the recipe for a cherry pie with one for shepherd's pie. It could go okay, but probably won't be edible, especially since the ingredients from each recipe were chosen randomly. Another possibility is that the hybrid individual will not have any disadvantages in terms of survival, but cannot produce its own viable offspring due to genetic incompatibilities. You might wonder how this works. Why can donkeys and horses produce mules, but mules are sterile? It is actually pretty simple. Horses have 64 chromosomes, and donkeys have 62 chromosomes. So mules get half from each parent and end up with 63 chromosomes, which means their gametes are turd emoji. The cell can't properly go through meiosis to create gametes because of that extra chromosome. When species have not had enough time for genetic incompatibilities to have occurred, hybrids are still possible. So a lion and a tiger that share a common ancestor 4 million years ago, the same as donkeys and horses, are still able to mate in captivity, though they would not run into one another in the wild. Polar bears occasionally make contact and mate with grizzly bears, which share a common ancestor 4.5 million years old. On a geological scale, this is not much time to create new species. Not everyone agrees with the biological species concept. It has some big problems. How do you identify asexual species like bacteria? How do you know two species cannot produce viable offspring? Are scientists supposed to start mating experiments in labs between every combination of species? That is really not practical. What do we do with fossils? And what about species that have not diverged? For example, a bird population may not split into two and yet change over time. If you could mate the species from 5 million years ago with its present form, would they be able to produce babies? This is called anagenesis, and we will never know. There are over 100 species concepts, and none are perfect. The ones that attract the most attention are the morphological species concept, which is the most practical because you can tell two species apart by looking at them, the ecological species concept, which is a proxy for mating ability, and the phylogenetic species concept, which calls separate clusters of individuals on a tree different species. A large part of my master's thesis focused on this last concept, which actually works pretty well a lot of the time. But in biology, there are always exceptions to the rule. Ponder this. There are salamanders in the genus Encetina in California that live around the Central Valley. Each population that is morphologically distinct can produce viable offspring with the population next to it. So it is one species, right? Nope. If you take two populations from across the valley and try to get them to mate, they are incompatible. This is a ring species complex. Perhaps your reaction is, well, that sucks. Species are so subjective. But think about it. Don't we expect species to sometimes be in the gray area? Species don't just wake up one day and realize that they can no longer mate with their cousins from another island. This actually supports the theory of evolution happening all the time. A geographic barrier that cuts a population in two creates potential species, which we call incipient species. Maybe the barrier is removed and they fuse back together, or maybe they remain separate. What do you think the result depends on? When we talk about speciation, we generally talk about allopatric speciation, which is when a geographical barrier splits a species in two. The event that causes this split may be the formation of the barrier, which is called a vicariant event, or it could be due to dispersal, where a handful of individuals manage to pass over the barrier. Let's look at some examples. Madagascar split from India 88 million years ago. The colored lines on this chart show the age of the common ancestor of taxa that live in both regions. The diving beetles, gentian plant, and mantellid frogs were present before the split, but the microhylid frogs and cichlid fishes share a younger common ancestor, which means they had to have dispersed from one region to the other. Your turn. Comparing the split between lemurs on Madagascar from mainland Africa to the maps. Was it more likely that they came to Madagascar by riding the continent as it slowly drifted away, or by swimming? By the way, lemurs probably did not literally swim across the Mozambique Channel, but instead got stuck on a large floating tree after a hurricane. This happens more often than you might expect. But geographical barriers are not necessary for speciation. Flies that live in the same forest but prefer different trees will only mate with individuals they come across that are also on the right tree. This can lead to sympatric speciation. As long as mating preferences are linked to habitat preferences, the result can be reproductive isolation. I like to think of allopatric as having two L's that separate one another, whereas sympatric has the root sim, like symphony, which means together. And sympatric speciation has the potential to lead to rapid evolution. Cichlids are just one example of this. Poison dart frogs are another. We call these speciation events adaptive radiations. Another form of speciation is polyploidy. This one is really cool. So a mistake happens in cell division, causing the organism to have the wrong number of chromosomes. And instead of being sterile like the mule, it is instantly reproductively isolated from its ancestors. This is actually really common in plant species. Giant strawberries are an example of a polyploid plant. Imagine you have two populations of a species. One, through non-disjunction, got an extra set of chromosomes. If the two come together to create a hybrid, and that hybrid mates with one of the parent populations, it is possible that a viable individual is created that can only mate with its own kind because it has a unique set of chromosomes. This individual is called an allopolyploid. So wait, if polyploidy can cause instant speciation, how quickly does speciation occur? Great question. I'm glad you asked. Darwin believed that evolution was slow. Others disagreed with his gradualism and felt that sometimes evolution can occur in short spurts. Stephen Jay Gould, whose work is so important that he appeared on The Simpsons, and whose essays are one of the finest pieces of scientific writing you will ever come across, came up with a controversial alternative that is now considered very sensible. Punctuated equilibrium is a theory that suggests most species change gradually or not at all for long periods of time. But then a large environmental change occurs and the population goes through rapid evolution. This is supported by what we see in the fossil record. I have one more concept to share with you about evolution. If you want to create a lot of change fast, one way to do it is to change the rate of development of certain body parts. Imagine your arms stopped growing when you were 3 years old, but the rest of you kept going. Instant T-Rex arms. We call this heterochrony. Tree-dwelling salamanders, for example, have toes that grow less, allowing for webbing that gives them better grip. Neoteny, or pedomorphosis, is when juvenile traits are retained as an adult and is one form of heterochrony. Cave salamanders evolved in this way because they retain their gills and light colors that are more useful in aquatic cave environments. Energy is also not wasted on larger eyes and toxins to avoid predation, since they don't have many predators in the caves they live. More complicated switches in the timing of development can lead to drastic alteration of the skeleton. For example, tiger salamanders that live in ponds that start to dry up can switch on developmental genes that change their body from a small, gilled insect eater into cannibalistic monsters. It has even been hypothesized that humans have undergone neoteny as a means of making our development slower, changing the shape of our skull and reducing the amount of hair on our bodies. Homeotic genes that are master controls for body part development can rapidly change the shape of a body segment, such that a leg becomes a claw or a swimming paddle. And antennae can be replaced by legs, or extra sets of wings can develop. The possibilities are endless for hopeful monsters that have unique traits that provide them with a higher fitness than their ancestors.